Hi guys, welcome to this video where I'm going to be talking you right through cell membrane structure for the OCR A-level specification. Now during this video we're going to look at lots of different things, the components that make up the phospholipid bilayer and also factors that can affect its permeability like ethanol, a type of alcohol, and temperature. Before we get started, don't forget to click the link in the description to access your free flashcards all about this topic. All you need to do is enter your details and I'll send them over to you immediately.
Cell membranes or biological membranes. This video is going to focus on the structure of the biological membrane and also things that can affect its permeability. We'll start with generally what is a biological membrane. Now, a biological membrane is a membrane that's found surrounding a cell entirely, which is separating it from its environment. So this part here on my diagram where it's shaded green, that's your cell surface membrane. But also within the cell, we're talking eukaryotic cells, we have all these membrane-bound organelles, which is separating the organelles from their cellular environment, from the cytoplasm, kind of compartmentalizes them into little areas where certain reactions take place. So, for example, the mitochondria, membrane-bound organelle, carries out respiration, which is separate to the Golgi apparatus, which again is separate to the endoplasmic reticulums. Lysosomes too, and the nucleus are membrane-bound organelles. Ribosomes aren't. Ribosomes are not membrane-bound organelles. They are organelles, but they don't have a membrane around them. So that's generally what a biological membrane is. In terms of its structure, we're looking at the phospholipid bilayer that forms a fluid mosaic model. The fluid mosaic model is a term that scientists use to describe the structure of the membrane. Fluid meaning the membrane is very dynamic, it's flexible, it's not static, it doesn't just sit rigidly still. And mosaic kind of describes the fact that it's not just composed of one type of molecule, there's many different ones in there. So just to reiterate that, fluid, it's actually the phospholipid molecules that make the membrane fluid, i.e. it's flexible, it moves a little bit. It tends to move, or the phospholipids move in a lateral direction, that means sideways, and that means the molecules embedded within the phospholipid bilayer, some of the proteins will also have a tendency to shift a bit in their positioning, always in a sideways movement though. And then in terms of the mosaic model, there's lots of different things within that phospholipid bilayer that makes it look a bit like a mosaic. We're talking really the proteins here, of which there are many different types, but there are other things there like cholesterol, which we're going to talk a bit about later on in the video. And we have glycoproteins and glycolipids, glyco meaning sugar, so sugar chains attached to protein, sugar chains attached to lipids. So you can see these in the diagram. We've got our glycolipid here. That's spelt wrong, isn't it? Glycolipid, that should say. And we've got our glycoprotein there. So that kind of just explains why scientists call it the fluid mosaic model. So we're going to look a little bit more at this idea of the bilayer, which is all to do with the main structures that make up the membrane, the biological membrane, which is the phospholipid. Different to a triglyceride. Triglycerides, if you recall from your first kind of area of biological molecules that you learn about, is a molecule made up of glycerol and 3 fatty acids. A phospholipid is slightly different because phospholipids have a phosphate and a glycerol head. This entire region is the head. And then there are 2 fatty acid tails, as opposed to 3, which you'd find in the triglycerides. Now, because of the different areas of the phospholipid, there are different properties, which is what makes the phospholipid form the bilayer. The head, made up of glycerol and this hydrophilic phosphate, basically means that they will attract, especially the phosphate area, to water. So where there is water, this phosphate group will attract towards it. There's a polarity. And that means that they are described as, well, the head is described as water loving because it will bond with water nearby, as opposed to the fatty acid tails, which repel any kind of water around them. So they would be described as hydrophobic or water hating. Now, those properties are really important because it's basically what makes the phospholipids form a bilayer, which forms our biological membranes. Here's a picture of our bilayer. So you can see we've got one layer of phospholipids on the top here, and then another layer of phospholipids on the bottom. And that's because in any kind of cell, the outside, the external environment, usually has a lot of water within it. So the fluids in which cells bathe are very watery. So there's a high water concentration, if you like. It's not just water, obviously, there's many other things in there. And then the cytoplasm of cells is also got a lot of water within it. So naturally, if you put a phospholipid in that situation around the edge of a cell, its tails are going to repel, or the tail is going to repel both watery areas, and the phosphate head is going to attract to the watery areas. And the way in which the phospholipid behaves is it ends up pairing up, and you have like a bilayer, two phospholipids, where the two tails end up repelling away from both watery environments, pointing inwardly, and the heads attract towards the watery environments, pointing outwardly. And that's what gives us this bilayer, this phospholipid bilayer that seals the cell, or seals the organelles within the cell from the cytoplasm. Now, all the different components that we see in this phospholipid bilayer have a function, have a role. They play a very important role in how the cell membrane ends up being this kind of partially permeable barrier to the environment in which the cell is situated, or it can help the cell be recognized by other cells or adhere to other cells, or even pick up receptor chemicals in cell signaling processes. So we're going to have a little bit of a breakdown of the different components and what they do. Now, we've kind of just talked about the phospholipids themselves. So they form the bilayer because of their hydrophilic, hydrophobic nature, and that ultimately is what makes the membranes partially permeable, because only substances that can dissolve and pass through fat, lipid, or lipid-soluble molecules can directly get through the phospholipid bilayer. Water-soluble molecules will be repelled from the tails of the fatty acids, so they won't be able to easily pass through. So this phospholipids makes the membrane partially permeable. Now, the proteins that are found within the membrane come in all different sorts of shapes and sizes and are positioned in different places. If the protein is seen spanning the entire width of the membrane, as we can see here in the diagram, of which there are two represented, we call these the integral or the intrinsic proteins. But what do? They actually enable substances that can't directly pass through the bilayer, or substances that are moving against their concentration gradient. They enable those substances to get through via channel or carrier proteins. So our channel protein here is typically a protein, an integral or an intrinsic protein, with literally a water-filled pore in the middle through which substances can travel that are water soluble. Carriers tend to have receptor sites in their structure which will bind to very specific shaped, complementary shaped molecules and move them across. And that can be through a facilitated diffusion process or even active transport if it's going against the concentration gradient. Channel proteins are used for facilitated diffusion, but not for active transport, whereas active transport is only carrier proteins, I should say, and then facilitated diffusion can use both. What about the extrinsic then, or the peripheral proteins? These are proteins which sit on the edges of the phospholipid bilayer, and it doesn't matter which edge, it can be the outer or the inner edge, depending upon their role. They are generally involved in things like cell signaling, which is how one cell can communicate with another cell. White blood cells are something you learn about a lot at A level, where you understand the process of cell signaling between different types of white blood cells using chemicals called cytokines or interleukins. So that could be the job of some of those extrinsic or peripheral proteins. They're involved in that process, or sometimes they're there to give the membrane a little bit of structural support. The glycoproteins, which means a chain of sugar attached to a protein, are also involved in recognition, cell recognition. I'm just going to remove that writing. And they can act as receptors to things like hormones, for example. And the glycolipids, quite similar in their role. These are the carbohydrate chains attached to the lipids, and they are also usually involved in this cell recognition and cell-to-cell adhesion. Just to go back on cell recognition, meaning that the cells are recognized by other cells in the body as self. And also cell-to-cell adhesion means when cells stick together. So it could be in the formation of tissues as an example. Now, cholesterol, we're going to talk a little bit more about cholesterol in the video a little bit later on. But just to give it the basic idea of what it's for, cholesterol is very important in controlling how fluid the membrane becomes. We don't want the membrane too rigid, otherwise things won't be able to get through. But at the same time, we don't want the membrane too fluid because too many unwanted things will get through. So the cholesterol has a very, very important job in maintaining the right amount of fluidity and hence stability of the membrane. And cholesterol works at different temperatures to ensure that the membrane is always maintained as this flexible but stable barrier between the inside and the outside of the cell. Okay, let's have a quick look at membrane permeability then. This will move us into the idea of things like cholesterol as well. Remember that partially permeable means that membranes will only allow certain molecules to pass through. The molecules that can pass through, well, it depends very much on how big they are, their size, their polarity, so do they have charge in their structure, and are they actually charged ions? So when I say polarity, I mean there's a little bit of positivity about them, a little bit of negativity, like water, for example. Charge would be a typical ion where it has an absolute charge, like a sodium ion. Now, the non-polar molecules, like oxygen and carbon dioxide, and they're very small, these generally will freely pass through the membrane by simple diffusion, as long as there is a concentration gradient, which allows that process to happen. So typically, these sorts of molecules would move between the phospholipid tails from one side of the membrane to the next, and because they're small, they can get through those gaps, and because they're non-polar, they're not repelled by the fatty acid tails. Molecules that are large won't generally get through those spaces, and if they're polar or they're relatively charged, they will be repelled by the phospholipid tail, so they won't pass through. Now, large charged polar molecules like glucose or amino acids or ions, because they can't just get through directly by simple diffusion, but the cell still needs them to get through the membrane for whatever process they're required for, they will get through using channel or the carrier proteins that we mentioned before. Factors affecting membrane structure. Now, there are two key factors that we learn about at A level that affect the membrane's structure and hence its permeability: temperature and solvent. So we're going to look at these both. In terms of temperature, now when the membrane starts to warm up, phospholipids are naturally going to gain more kinetic energy. The phospholipid tails are going to start to move a little bit more. And as a consequence of them moving around more, it makes the membrane more fluid and more permeable. So as they move, things are more likely to be able to get through the gaps between the phospholipid tails. Now, if the temperature keeps rising, the membrane will become more and more fluid and more permeable. And this is particularly the case if there are many unsaturated fatty acids which form part of those phospholipid tails. We'll talk about that on the next slide. If temperature was to increase way too much, and that can be detrimental to the membrane, the channel and the carrier proteins, just like any kind of protein, like enzymes that you learn about, they can become denatured due to extreme temperatures where bonds break, their shape literally breaks down, and they lose their structure. And at that point, you end up with literal gaps in the membrane, and the membrane becomes extremely permeable and is no longer able to control what's moving in and out. Now, low temperatures are also going to affect membranes fluidity. We're talking extreme low temperatures again, guys. We're not just talking about just when you're a bit cold. If a membrane goes below 0 degrees C, then the water that's always moving through membranes by osmosis is going to freeze and form ice crystals. When the membrane thaws, those ice crystals, because they've expanded, water expands when it freezes, then it's going to pierce the membrane and literally break the membrane down. It's going to make the membrane fall apart. So having ice crystals forming in the membrane is completely detrimental to the membrane structure. We don't want that happening either. Let's go back to the idea of saturated and unsaturated fatty acids, a little bit of chemistry. Now, a saturated fatty acid is where the hydrocarbon chain has no double bonds between the carbons. So the carbons are fully bonded to a maximum amount of hydrogen possible. So that's why we describe them as saturated fatty acids. And that has quite a big effect on how the fatty acids behave at different temperatures, for example. Unsaturated fatty acids are the opposite. You will find some double bonds between some of the carbons, which means less hydrogen is able to bond with those carbons because carbon always forms 4 bonds around it. So if it's forming a double bond, that reduces its capacity to form a bond with more hydrogen. Just finish that off so it makes chemical sense. So that would be an unsaturated fatty acid. Now, unsaturated fatty acids tend to have kinks in their tail, in their hydrocarbon chain where the double bond is. So a saturated fatty acid in a phospholipid they'd be particularly straight fatty acids, whereas in a phospholipid that had unsaturated fatty acids, you might see like a kink in the chain, like so. Bit of a rubbish diagram, but just to try and explain that you get these little kinks in the tail, in the fatty acid tail. So if you have more saturated fatty acids in the membrane, what the impact of that is the membrane actually becomes less fluid at low temperatures because saturated fatty acids at low temperatures tend to become quite solidified or compressed. And when they are compressed, they're going to move less. And if they move less, then less things are going to get through between the fatty acid tails. Therefore, membrane fluidity is reduced at low temperatures due to these fatty acids becoming compressed if they're saturated. Now, unsaturated fatty acids also enable a degree of control over fluidity. Now, at low temperatures, the unsaturated fatty acids are there to ensure the membrane doesn't become too solidified. You don't want it to lose its flexibility completely. So what the unsaturated fatty acids do is they help to maintain the fluidity. Membranes will have a combination of both types of fatty acid and they both do their job. So the saturated fatty acids are there to kind of stop too much fluidity. Unsaturated fatty acids maintain a bit of fluidity because they don't become as compressed at the low temperatures. They therefore enable the phospholipid tails where these exist to keep a little bit of flexibility and things can therefore move through. So it's kind of like a happy medium of having the 2 types of fatty acids within the phospholipid tails. Now, solvents can have a huge impact on membrane permeability, which we don't want. This is a bad thing. We know that phospholipid bilayers form because of the way phospholipids behave in water. They form the bilayer, the heads are on the outside of the bilayer and the tails are on the inside of the bilayer. Remember, that's because water is polar and it's a solvent. So there's this like beautiful relationship that water and phospholipids have with each other. And this ensures, that the phospholipid bilayer is maintained, it's intact all the time, unless a solvent comes along. Now, a solvent like alcohol ethanol typically, alcohol generally will insert itself into the lipid bilayer, the phospholipid bilayer in between the phospholipid tails, and it breaks the bonds that keep the orderly kind of pattern and arrangement of the phospholipids with each other. It basically interferes with the interactions between the phospholipids and disrupts their kind of arrangement that keeps them at a happy fluidity. And as a consequence of that, alcohol makes the fluidity of the membrane and its flexibility go up. It becomes too fluid, too flexible, and therefore becomes leaky, making it more permeable to small molecules and ions. So it disrupts the membrane in a bad way. Now, cholesterol, just to go back to cholesterol, which is a type of sterol, very important because it helps to stabilize membranes. So it ensures the membrane is in a perfect kind of fluid arrangement. So we have enough fluidity going on without the membrane being too rigid, but we also don't want it too fluid. So how does cholesterol play a part in this? Now, cholesterol interacts with fatty acid tails. The more it interacts, the less fluid. So what we're saying is, here's some cholesterol. If the cholesterol is sticking to the fatty acid tails, it stops them moving around as much and makes the membrane less fluid. But if the cholesterol is not sticking to the phospholipid tails, then the tails are free to move more. At low temperatures, cholesterol becomes a little bit more solid, so it's less likely to interact with the phospholipid tails, and therefore the phospholipid tails are likely to be quite loose. They're not packed really tightly, and that enables the membrane to be pretty fluid. The more cholesterol, the more fluid in the membrane at low temperatures. However, if we were to look at what happens if the membrane gets a little bit too cold or a little bit too warm, we have to have an understanding that cholesterol is also really good at ensuring the membrane doesn't get too interfered with by those temperature changes. So when cells are a little bit cold, as we just looked at on the previous slide, the cholesterol is relatively solid. It doesn't interact very much with the phospholipid tails. They don't pack too tightly together, and the membrane is relatively fluid, so it maintains a little bit of flexibility and movement. As the body warms up, cholesterol can reduce the fluidity from becoming too much. So if obviously phospholipids get too warm, they gain kinetic energy and become very, very, very fluid flexible, and that can cause the membrane to become a little bit too permeable. But cholesterol also prevents that from happening. As the cholesterol warms up, it becomes a little bit stickier, and as a consequence, it interacts more with the hydrophilic tails, stops them moving around too much, and makes the membrane stronger and more stable, preventing it becoming too fluid at those warmer temperatures. So cholesterol is like the membrane's perfect friend. It helps it in both extremes, when the membranes or the cells are a little bit cold or a little bit warm.
Guys, if you want even more, then don't forget to check out the Biology My Resources on the My EdSpace website. We have video explainers, practice questions, and video solutions. If you want to continue learning with me, then you can sign up for our biology courses. You will get 2 lessons a week, workbooks, exam-style homeworks, and video solutions. Just click the link in the description. If you like this video, then don't forget to subscribe, like this video, and put in the comments any other topics that you'd like me to cover. We do actually read them. So I'll see you in the next one.